रोशनी रोशनी उम्मीद है तामीर दाए रोशनी उम्मीद है
Bismillahirrahmanirrahim. Assalamu alaikum and good morning everyone. My dear students, our today's topic is from unit number 3, biodiversity. That is viruses as unique particles. Today I'll teach you about the most amazing and interesting topic of biology from unit number 3, biodiversity. And topic is viruses unique particles. A virus is a submicroscopic infectious agent that replicates only inside the living cell of an organism. Viruses can infect all types of life forms, from animals and plants to microorganisms, including bacteria and archaea. In our today's video, we will discuss about the definition, characteristic of viruses. We will also discuss about the structure of different viruses like DNA virus and RNA virus.
Morphology and structure of viruses. Let's start with definition of viruses. Virus is a smallest infectious particle, unicellular, obligate intracellular parasite. So virus is the smallest infectious particle. It can cause various diseases to human beings, plants and animals. It is unicellular. It has a single cell, obligate intracellular parasite, that is, it multiplies in the host cell. Let's see some important characters of viruses. Now, characters of viruses. In characters of viruses, we are going to study size of viruses, shapes of viruses, structure of viruses, and in structure of viruses, we are going to see structure of capsid, nucleic acid, and envelope. So, let's start with it. Let's start from size of viruses. Viruses are smaller than bacteria and they are filtrable. So, these viruses can easily filter through bacteriological filters. Size of viruses is measured by using ultrafiltration, ultracentrifugation and electron microscopy. So, these are the three methods that are used for measurement of a virus. Size of viruses ranges from 20 nanometer to 300 nanometer. Example, size of parvovirus is 20 nanometer and it is the smallest virus, whereas size of pox virus is 300 nanometer and it is the largest virus. Let's see shape of viruses. There are different shapes of viruses. Mostly all animal viruses are roughly spherical in shape, but there are also different shapes of viruses. So maximum all animal viruses are spherical in shape, but there are some viruses that show variety of shapes. So let's see different shapes of viruses and their examples. TMV, that is tobacco mosaic virus, is rod shape. Pox virus is brick shape. Rabies virus is bullet shape. Coronavirus is petal shape. Rotavirus is wheel shape. And astrovirus is star shape. Ebola virus is filamentous in shape. So you can see variety of shape. So first one is roughly spherical, rod shape, brick shape, bullet shape, petal shape, wheel shape, star shape and filamentous in shape. So this was about the shapes of viruses. Let's see structure of virus. Virus is mainly made up of two components, and that is nucleic acid and capsid. But some viruses may show presence of envelope. So the two most important components of virus are nucleic acid and capsid, but some viruses show presence of envelope. Nucleic acid is enclosed with a protein coat called as a capsid. So nucleic acid is surrounded by a protein coat and this protein coat is called as a capsid, and this capsid may be surrounded by a lipoprotein layer called as an envelope. So in the center nucleic acid is present. Surrounding to nucleic acid, a protein layer called as capsid is present, and surrounding to capsid, there may be a presence of lipoprotein layer called as a envelope. Envelope is derived from host cell membrane, so chemical structure of envelope of viruses is similar to the cell membrane of host cell. Let's see capsid. Capsid is made up of a protein coat called as capsomers. The chemical unit of capsomers are polypeptide molecules. Capsid show two types of symmetry, and that are icosahedral symmetry and helical symmetry. Now, icosahedral symmetry is made up of 12 vertices and 20 corners, as well as it shows two types of capsomers, that is pentons and hexons. Now, the 12 vertices are made up of pentagonal capsomers, that is pentons, and the number of pentons are fixed, that is 12. The sides are made up of hexagonal capsomers, that is hexons, and the number of hexons may vary. Now, in helical structure, here nucleic acid and capsomers are bound together to form a helical structure. So, here nucleic acid and capsomers get coiled to each other, they are bound together, and it may get coiled or itself on itself, or the helical structure may be rigid. So, these nucleic acid and capsomers may get coiled to itself, or the helical structure may remain rigid, that is, it may show a rod shape. Now, some viruses may show complex symmetry. Now, let's see the function of capsid. Capsid protects nucleic acid from nucleases and other environmental factors. Now, role of nucleases is to disintegrate nucleic acid. So, here the function of capsid is to protect nucleic acid from nucleases and other environmental factors such as heat, temperature, water. Now, capsid introduce viral nucleic acid into host cell by absorption mechanism. This is the second most important function of capsid. It introduces viral genome in the host cell. Now let's see nucleic acid. Viral cell have only one type of genetic material, that is, it may contain either DNA or RNA as a genetic material. So any virus cell may contain only one type of genetic material. It may contain either DNA or RNA, but not both. Viruses do not contain the enzymes that are important for replication and multiplication of viruses. So, the enzymes which are necessary for replication and multiplication are not synthesized by viruses. So, virus cells are completely dependent on host cell for its multiplication and replication. Now, let's see DNA viruses. Now, here it is clear that there are two types of viruses, that is DNA viruses and RNA virus. So, let's see DNA virus. All DNA viruses are double-stranded except parvovirus. Parvovirus has single-stranded DNA. So, all DNA molecules, all DNA viruses are double-stranded in nature. Now, let's see the examples of DNA viruses. Vaccinia virus, human papilloma virus, parvovirus, herpes simplex virus, hepatitis B virus, and adenovirus. So, these are some examples of DNA virus. Further, RNA virus. All RNA viruses have a single copy of single-stranded unsegmented RNA. And examples of RNA virus are mumps virus, measles virus, influenza virus, influenza A, B and C, rotavirus, reovirus, human T lymphotropic virus, and HIV. Further, let's see about envelope. Envelope is the outermost covering of viruses. The viruses that contain envelope are called as envelope viruses. The viruses in which envelope is absent are called as non-envelope virus. So, on the basis of envelope, there are two types of viruses, that is envelope viruses that show presence of envelope and non-envelope viruses that show absence of envelope. Envelope of viruses is made up of lipoprotein molecules, and this envelope is derived from host cell membrane during lysis of cell. So, when a viral cell carry out its multiplication, its replication, at the end stage when the virus cell undergo lysis of that cell, It takes some part of cell membrane and forms an envelope, and this envelope is made up of lipoprotein molecules. These lipoprotein molecules are derived obviously from the host cell membrane. The chemical composition of envelope is similar to the host cell membrane as the envelope is derived from the host cell. So, this was all about viruses. Thank you.
Today's assignment: learn and write these 5 questions. 1. Discuss shape of the viruses. 2. Describe structure of the virus. 3. Define envelope. 4. Define symmetry and its two types. 5th. Define DNA virus and give examples. Best of luck. Take care and Khuda Hafiz.